So now let's talk about meiosis. First, let's start with our drawing of a cell. And here are our chromosomes, pink for mom and green for dad. And just like in mitosis, the first thing we have to do is replicate the DNA. So now we have a pair of homologous chromosomes going into prophase 1. But in prophase 1, something special happens called synapsis. This is where our sister chromatids pair up in what is called a tetrad, and this allows them to cross over or exchange genetic information between non-sister chromatids. So now we have 4 daughter chromatids, all with different genetic information. Next, during metaphase 1, we get alignment of the chromosomes on either side of the metaphase plate. And each pair of chromosomes can independently assort, which means that mom's chromosome can be on one side of the metaphase plate, dad's on the other, and this can vary for each pair of homologous chromosomes. Next, during anaphase 1, our homologous chromosomes each are drawn to opposite poles of the cell. And during telophase 1, our mitotic spindle breaks down and our nuclei partially reform. However, remember that this does not have to go to completion during meiosis 1. Next, we enter into interkinesis, which is a short rest phase before meiosis 2. And keep in mind, the DNA is not replicated again before meiosis 2. So now, going into meiosis 2, we have a pair of daughter cells from meiosis 1, and each contains 1 chromosome from each homologous pair. Now, during metaphase 2, our homologous chromosomes line up along the metaphase plate similarly to how they do in mitosis. So that during anaphase 2, we get separation of the sister chromatids, and they are pulled to opposite poles of the cell. During telophase 2, now we get the dissolution of the mitotic spindle again, and reformation of our nuclei around our now daughter chromosomes. And finally, we get cytokinesis. And this results in the production of 4 haploid daughter cells, or gametes. And that is the process of meiosis.